Hello, I'm Irene Iheer of CDRH's Office of Communication and Education. Welcome to the FDA's 30th in a series of virtual town hall meetings to help answer technical questions about the development and validation of tests for SARS-CoV-2 during the public health emergency. Today, Timothy Stenzel, Director of the Office of In Vitro Diagnostics and Radiological Health in the Office of Product Evaluation and Quality, and Toby Lowe, Associate Director of the Office of In Vitro Diagnostics and Radiological Health, both from CDRH, will provide a brief update. Following opening remarks, we will open the line for your questions related to today's discussion. Please remember that we are not able to respond to questions about specific submissions that might be under review. Now, I give you Toby.
Thanks, Irene. Hi, everyone. Thanks for joining us again this week. I've got a couple of announcements that I wanted to share with you, and then we can open up the line for questions. So the first thing I wanted to mention is last week, in case folks didn't see it, we put out a letter to healthcare providers on recommendations on providing clear instructions to patients who self-collect an anterior nares nasal sample in a healthcare setting for SARS-CoV-2 testing. So that walked through the importance of having appropriate directions for patients who are self-collecting so that we make sure that they get a good sample prior to that sample being sent into the lab. And we link in that letter to healthcare providers to a couple of examples of written instructions that can be used that are available for public use. So providers can use those if there are not other instructions that they already have available. So we encourage folks to take a look at that. Along with that, on the same day last week, we also updated some of the FAQs related to swabs, both in the testing supply FAQs section and the 3D printed swabs FAQ section on the FAQ page. So take a look there for some updated information. And then yesterday, we issued an immediately in effect guidance on titled The Enforcement Policy for Modifications to FDA-Cleared Molecular Influenza and RSV Tests During the COVID-19 Public Health Emergency. We put this policy into place to help with the potential for shortages during the upcoming flu season, since a lot of the flu and RSV tests use the same components as many of the SARS-CoV-2 molecular assays. So this policy will help expand access to certain FDA-cleared molecular tests intended for detection and identification of flu viruses, and it includes those influenza tests that also detect and identify RSV. So that was issued yesterday and is posted up on our website as well. And with those updates, we can go ahead and open the line for questions. Thanks.
Operator will now take questions.
If you would like to ask a question, press star 1 from your phone, unmute your line, and speak your name clearly when prompted. Again, if you would like to ask a question, press star 1. One moment as we wait for any questions. There are currently no questions in queue.
Operator, can you get scripting one more time? I'm sure that we have questions folks are probably trying to get into the queue.
If you would like to ask a question, press star 1 from your phone, unmute your line, and speak your name clearly when prompted. Again, if you would like to ask a question, press star 1. One moment as we wait for any questions. Our first question comes from Adam Beebe. Your line is open.
Hi, yes. First off, thanks for everything you guys do. Just a quick question. What are some of the circumstances that might allow for one company to receive EUA approval after, say, 6 weeks versus other companies that have been waiting for well over 100 days? Thank you.
So we do have a triage system, Adam. If you think that triage system, or you think where you are in that triage is not appropriate for your developed test, if you haven't already done so, please communicate with us. I know we have some sponsor-specific, developer-specific conversations going on, and we'll continue those to make sure that they're properly triaged. And we clearly have stated recently that those EUA submissions that require EUA authorization before they can be implemented, like any sort of home collection, home tests, of course, point-of-care tests. They can't be point-of-care unless we've reviewed and authorized and deemed it as point-of-care. And then, in addition, those things that substantially add to the ability for our national response in terms of throughput, high throughput testing. The other thing is, those submissions that are incomplete may not be triaged higher on the list. So every submission in my direction has a contact within 2 weeks in the office. So if you want to check that your submission is complete, you can ask that contact. You can also ask that contact to double-check your triage status and make sure that they understand the features of your test and how it might be a higher priority. But, from the beginning, we've received over 2,400 applications, EUA applications, as of last month. We've authorized going on 300 tests. We've authorized over 200 amendments to EUA authorized tests, and we've denied something on the order of 100 tests. So we are making decisions as soon as we can. Those who have serology tests that are amenable for testing at NCI, we are encouraging all developers to submit their tests to NCI. And we are, there is still a backlog, although of those that we've prioritized at NCI, the backlog is shrinking. And then once we get data from NCI, we try to make our decision as quickly as possible. And obviously we share the NCI data that we receive with the sponsor. So hopefully that helps. And Toby, I don't know if you want to add anything else.
No, I think you covered it. Thanks.
We will take our next question.
Our next question comes from Laura Mazzola. Your line is now open.
Hi, thank you. Laura Mazzola with Halteres Associates. I have a question on the EUA template for self-collection kits. The prior template was coupled to an EUA test, and I'm curious now if there's going to be a revision of that template specifically for use with an LDT, which no longer requires emergency use authorization, or what are your recommendations for sort of in-house kits for self-collection?
Yeah, that's top of mind and top of list for us to provide some more clarity. And Toby knows that I've assigned her to help with that. Yeah, no, we would encourage that if you can use an EUA authorized collection device that fits with your test and under the authorization of that collection device, so that would be a great way to go. If you're not got a home collection device that has been authorized, then yes, it's time. It's something that we want to engage in a conversation with you. Of course, the Gates Foundation and others have done significant work in this area and have provided writer reference for some data and some information, and so we would like to leverage as much of that as possible when we look to authorize additional home collection. But you bring up a great point, and it's one that we're working through now. So I can't give any high-level anything other than that as far as details go now. So do send us an email at our template or talk to your reviewer about this particular thing, and we'll work through these things on a case-by-case basis. Toby, you have anything to add to that?
I think that's a good overview of where we are. I think emailing the mailbox about your particular situation is a good first step at this point, so that we can work through any issues with you directly.
Okay, great. Thanks very much for that.
Our next question comes from Lonnie Edelman.
Edelman for iAssay. I've called in once before. We have a cloud-connected device that is an open platform for reading scripts and recording the data automatically. A couple of weeks ago, Tim, you made a comment, and we're still kind of confused. You had said that developers don't have to use an instrument that's been prior authorized. They can use whatever they want as long as it works. So this is kind of different than what our prior understanding was. We sent in an email. We got an email back. It was kind of cryptic to us. So this would open up things for us if we could actually get to potential partners, either starting at the end user and doing some testing, presenting the data. So I wanted to find out, is it really a change in policy where we wouldn't have to put our device onto somebody else's EUA in order to use it for testing?
Yeah, I think you're talking about a device that can read a lateral flow strip, primarily a serology strip.
Serology or antigen, yes, sir, that's correct.
Right. So certainly all the EUA authorized devices are posted on our FDA website, and if they don't already have a device, or if they're interested in adding a new device, you can work with them. We are working on a template update, and I hope it gets cleared. We would like to open it up and make this as easy as possible for a scanning device to be used, and we're just waiting for the final sign off on that template update to provide some specifics on that. In the interim, we can provide our up-to-date recommendations about how to do that. It sounds like you've already tried that, so if you and if I was included on the email, I apologize. I get inundated with emails, but
yeah,
I do want to open up that. I do want to open up this pathway, so you can reply and say you're a little confused, and can you please copy Dr. Stenzel and get his input on this, because I know I have some thoughts about this. I've reviewed some of the updates, and I'm happy with it. So we can provide elements of that current thinking, not a completed template, but elements of it that we're comfortable giving you some recommendations on how to move forward.
What's the name of the template?
Well, let's see. I don't remember if this is an update to the serology template or an update to the direct antigen template, or both. I forget.
Yeah,
And
I believe we're in the process of updating both of those.
right. So I just don't remember where this additional information on how to work somewhat more flexibly for providing an image system. But I wanted, and my desire was to make this as easy as possible for all developers and as easy as possible for the paperwork end of what we do at the FDA to add something like your product to a test and have it be able to be used and EUA authorized.
Yes, that's great. I mean,
if I could just add, I think, I'm recalling, although I don't have it in front of me, I'm recalling the email question that you sent in as well. And I think, there may have been a little bit of confusion around, us stating that you can use any instrument as long as it works. The intent there was just to make it clear that the instrument didn't need to have prior authorization before the emergency, and that it didn't need to be a device that, like a platform that we've seen before. You can bring in new platforms that we've never seen before during the emergency for an EUA. So as long as it works, we would be authorizing it under an EUA, just like we would with, with something that we're more familiar with.
Okay, so that clarifies. So we're very interested in getting this in the hands of people. It's going to solve a lot of problems. We can prove it. We've already have. And so this template will be updated. Sounds like it'll be great. I don't want to monopolize time. One question we haven't been able to figure out, how do we get this NCI panel for testing? We've looked, we've dug. How do we get it so we can do testing with it?
Yeah. So unfortunately, it's sourcing the serology samples in sufficient volume to support the NCI testing has been challenging enough. And though it was our desire early on in the process to source enough sample that we could support the testing at NCI and make the panel available to developers who don't have a device that's amenable for testing at NCI, but because of the challenges in sourcing that material in sufficient numbers and quantity and volumes, we've been unable to meet that need at this point.
So if we want to try to get it and get in the queue, who do we talk to?
Well, the only thing that they're doing right now is they are accepting kits for primarily, some ELISAs, but primarily lateral flow devices to get in the queue for testing at the NCI. So a device that scans such things wouldn't be something that we would test unless it already comes in validated with a specific device, and that's part of the kit already. It's being submitted to.
Okay.
All right.
All right. Thank you.
Uh-huh.
Our next question comes from Kimberly Zunker. Your line is now open.
Thank you. My question is about the new FAQ surrounding laboratory-developed test EUAs, stating that they will not be reviewed. I'm wondering if laboratories are still required to submit their EUA applications within 15 days of validation if they're not going to be reviewed.
Debbie, how do you want to handle that question?
That's an interesting question. So the guidance document, I believe, is what you're referring to. That was issued well before the HHS statement and the FAQ that you're referring to. And as I'm sure you're aware, guidance documents are non-binding. So the recommendations in the guidance were our recommendations at the time that was issued. And we, as we've now said that we are generally not prioritizing review of LDTs, and HHS has said that that FDA will not require pre-market review of LDTs. The current FAQ is, I would say, more current than the portion of the guidance that you're referring to. Tim, I don't know if you want to add to that at all.
No, but I think we've heard some, and we are working on some additional clarification in the FAQs, and this is a good question for us to potentially address. As it appears, there's a discordance between existing guidance and the FAQ. So thank you for bringing that to our attention.
Yes, absolutely.
Thank you.
Our next question comes from Ashley Douglas. Your line is now open.
Hi there. Could you provide a little clarity on if you have an existing EUA for a high throughput capacity, and you want to add flu AZ, if you're recommending an amendment to an existing EUA, and is that a possible pathway?
That typically would be a new submission, a new test, and not an update, unless you're going to sunset the other test. But even so, it's probably for our records best to keep that as a separate submission. And we've provided some pretty good recommendation in our molecular template for that. I believe also we've authorized the first antigen, indirect antigen test that's a panel, but I don't know that our current template is updated for a panel test. That's obviously in the works as well, to provide updated recommendation. But if you have a direct antigen test that you want to move to a panel, for those listening, just approach us and ask for our current recommendations on how to do that. So, so short answer is I think that's best handled by a new submission, since a new assay and a new record of validation, because even for SARS, we would want to make sure that SARS is not impacted by the additional panel members that analytes that you're adding.
And I would just add that you can reference your previous submission. So if there are elements of your previous submission that are still relevant, you would be able to reference that instead of providing it new.
Yeah,
Okay.
Good point, Toby. Although a lot changes, right? A lot changes with that.
Okay, thank you.
Our next question comes from Robert DiTilio. Your line is now open.
Hi, Tim. Thanks for this. And actually, my question was surrounding the self-testing, so it's been answered. But it's since I'm on here, hello, it's fascinating that you have had 2,400 submissions. How many staff do you have in each one of the three groups, approximately?
Not enough.
You don't have enough. No, there's not enough people in the government. I'm just wondering.
Yeah, and that includes both pre-EUAs and EUAs. I think we have over 1,000 prior to our LDT decision. We had over 1,000 active EUA submissions still. And so we're working as hard as we began the year. The virology branch within DMD was about 25 people. We subsequently doubled it with direct reviewers, bringing in other people from the office, from the OPEC, and from the agency. We've hired about 60 people in the office so far this year. And then we are now adding additional staff from other areas of the office. So our direct reviewer numbers will be approximately 100 now, with a large support staff behind them, assisting in every way. So it is it has been very challenging. It is why we have utilized the notification pathway for nearly every type of submission except for some, like home collection and home tests. And for obviously for point of care decisions. So we've done our best to stay up and been flexible with our recommendations and guidances to deal with the volume. So thanks for that question.
Fascinating. Keep up the good work. Thanks very much.
Thank you.
Our next question comes from Griffin Soriano. Your line is now open.
Hello. Thank you for taking my question. Just curious if the FDA has any updated position on the off-label use of antigen testing for asymptomatic screening, given how forceful HHS was in requiring Nevada to resume the use of antigen testing in nursing homes for that purpose. Does the FDA have a position on this and this off-label use, and more importantly, are you aware of any studies to measure the performance of the antigen testing in the asymptomatic population?
Yeah, the data we've received, and I can't go into all of it because much of it is confidential, is mixed. Some studies seem to say that the viral levels, at least as determined by cycle thresholds, which is an imperfect way of determining virus thresholds or virus levels, are similar in some cases, in others very different. And we don't understand why that is. So, and when they're different, the asymptomatic population usually has a lower viral load than the symptomatic population, and that could be from a variety of reasons as well. And we make decisions based on data, and since the data are unclear, the science is unclear now, it's difficult to sort of make blanket recommendations about this. So we have asked developers if they want to claim for screening specifically asymptomatic patients that they come in with data. We've given a large number of options for doing that. We've obviously authorized some already, and we're very interested in authorizing others. And typically, when we ask for a small number of patients who are known to be asymptomatic and do a comparison study from the test that's being submitted with another high sensitivity EUA authorized test. And then there's also the ability to use bank samples. There's also the ability to look at for where it's applicable to historical data, where you have data from both types of patients and it shows comparable cycle thresholds. I'm trying to think of the other ways that we've been allowing those claims. But we very clearly have stated in our FAQs and other communications that that most of the IUs that we writing in the IU and the instructions for use and the indications for use, we've clearly stated those suspected of COVID-19, and that can be people who have symptoms, but it can also be many patients who are without symptoms but are known to have an exposure, where you want to make sure that they aren't carrying the virus. And then there's purely sort of the screening. You've got, you just want to know, say, in some setting where you want to know or if anybody in that population, and you want to return individual results to them, that screening, then, you can come in with a submission for that claim if a submission is required, if you're primarily a kit. But also, we've very clearly said that clinicians can order this off-label. There were some other challenges at the federal level with CLIA and CMS. They have issued a statement, a FAQ, that says you can do this, and you can get reimbursed, and you can report, and you're not going to get dinged by a CLIA inspection. The specific issue that I know was seen in Nevada and many other nursing homes recently, and certainly whenever there's a complaint about, say, a false positive, we look at those complaints and we do an investigation as required and determine whether or not there's a device issue. But I think the challenge is that in those populations, it doesn't even matter what particular test, whether it's authorized for asymptomatic testing or not. No test is perfect. No test is 100% specific. And so if you have, that includes molecular tests, by the way. Those are not 100% specific. They may be very high, maybe even higher than direct antigen tests, but they're not 100% specific. That's sort of impossible to reach. And so when you have a very low incident population of positive results, the chance of having a false positive is very high. And therefore, if it matters whether that's a true positive or not, whether it's in somebody who's staffing the nursing home, or it's a healthcare worker in another environment, or it's an actual patient that you're interested in knowing, when the population you're looking at is very low incidence, you want to double check that result. So I would send it off for a different orthogonal test. This is very clearly stated in current CDC FAQs and guidance, and it applies not just to direct antigen tests, but also to all tests. And just to go back and just talk about this a little bit. So if you have a test that is 99% specific, that is a fixed number. It doesn't matter what the incidence is in the population. It means on average 1 out of every 100 negative patients will test positive with that test. And I think most people would agree that a 99% specific test is pretty high. So if you suddenly have a population that has significantly less than 1% incidence of true positive results, it means that there will be more false positives than true positives. And I think that's where what Nevada saw, and it, and dealing with that, it can be a challenge, granted, but it is nothing wrong with the test. It's nothing wrong with the test method. It's nothing wrong with what the test is labeled to do, given that the federal government, CMS, CDC, and the FDA have said it's okay to use a device off-label when you have a clinician prescribe this test. So that's a very long answer, but I think it's very important. The testing that is able to be done in these populations is very important because my understanding of the Nevada data and other data is that you are identifying true positives that you in a faster fashion than you might otherwise. So I personally view that as a very important public health tool. And Toby, I don't know if you have anything to add to that.
I think you covered all of it. Yep.
Great, thank you.
Uh-huh.
Our next question comes from Tom Slavic. Your line is now open.
Thanks. I'd like to confirm my understanding of the options for a company that wishes to market VTM. There seem to be 3 separate ways to go. First is that the VTM notification pathway is sufficient to be able to market swab collection kits that include a transport tube with VTM. The second is that there's an EUA template for saliva collection kits to be used for SARS-CoV-2 only. And the third is that a 510K is required for potential broader use of a saliva collection kit. Is that a correct understanding?
That sounds pretty good to me, but Toby has been focusing on this a little bit more than me. So, Toby, you want to address that?
Sure. So yeah, what you've outlined sounds pretty much correct. So when you're saying that there's a EUA for saliva collection devices, we don't actually have a template specific to saliva collection devices. We have authorized EUAs for 2, I believe, saliva collection devices at this point. Those are standalone collection devices. They are not home collection kits. We have also authorized several home collection kits that include, there are some that are saliva and some that are nasal swabs. And for VTM, separately, we do have the immediately in effect guidance. So many swabs are Class 1 exempt if it's just the swab, and the transport media is what bumps it up to meeting the 510K. So generally, those do need 510K, and we are not generally looking at EUAs for transport media specifically because we do have this guidance document out with the policy that allows for enforcement discretion for the 510Ks that are required for that transport media. So if you want to use to market transport media during the public health emergency, you can take a look at that policy and the guidance, and that is not limited to SARS-CoV-2 testing. That is for the distribution of transport media during the public health emergency, but it is for more broad testing, for molecular testing, I believe. I'd have to look at the exact language in the guidance. So that hopefully that does clarify a little bit there for you.
Yeah, Toby, good point about the VTM guidance. Some people have thought it only applies to SARS, and it doesn't. It is broad in the pandemic. And I don't know if the new IE for flu and other respiratory pathogens, you want to discuss its relevance to this question at all either. You can just check that all as well.
Sure. So the new guidance about flu is sort of the flip side of the transport media guidance. So the transport media guidance talks about the manufacturers of the transport media and their need for a 510K and the ability to use this policy to distribute without a 510K during the emergency. And then the flu guidance talks about the regulatory requirements for the manufacturer of the flu tests themselves and the ability for them to use this policy to expand the use of their flu test to use these other transport media that may not have been included in their 510K initially.
Okay, thanks. I think you confused me a little bit. If someone wants to market a swab collection kit that includes a transport tube with VTM, is the notification pathway sufficient, or do they have to apply for 510K? That's the hard part I've been trying to...
The notification pathway would be available to them. Sorry that I was confusing there.
Okay, great. Thank you very much for this clarification.
Sure.
Our next question comes from Troy Moore. Your line is now open.
Hi, thank you for taking my call. I have a two-part question regarding the announcement around the LDTs last week. There wasn't a time, so we have a submission in, and I was just curious if there was a greater clarification on the timing of receiving notifications of whether the review is going to proceed or be declined based on a determination of an LDT status.
Yeah, so we've reached out to a number of LDT developers and let them know already, but that process is not complete. And if you want to check on it, you can send an email to your contact, whether that's a contact within the office or a reviewer, or you can use our template's email address as well. And we'll attempt, we're engaging in dialogue with some to make that determination, and that's on a case-by-case basis, so that may take us a little bit more time to work through that.
Yeah, and I guess that was the second part of my question was, we have a pool-based methodology, 25 samples or more, using a next-generation sequencing readout, and I didn't know if there's a stratification on, things that would clearly fall in the LDT bucket and things that would not, or if you guys have any clarification on that?
Yeah, we're doing our best right now, and we're working through that very important question. And as I said earlier, I think we're going to work on some additional FAQs to post to make this a little bit more clear and transparent. But for now, I think it's best, this be handled on a case-by-case basis so we can give you the very best feedback and recommendations and answers that we can tailored to your use.
Okay, certainly heard you loud and clear about the overwhelming number of submissions that you guys have received. So I really appreciate the effort you guys are putting into this.
So, well, these kind of questions we try to handle very quickly. So this is not part of a submission review. If you're coming in and you say, do we fall under this new FAQ for LDTs, and you're going to choose not to review it. That's a straightforward, seemingly straightforward question that we want to respond to as quickly as possible, and that's what I've directed the office to do. And we are engaging obviously with numerous discussions. So that's not something that you're going to that I'm expecting that you're going to have to wait a long time for a response. We want to get back to you as quickly as possible on that, and that's what we're doing. And Toby's thankfully is heading up the response team here and making sure that that we're getting back to developers as soon as possible.
Yeah, those types of questions we usually get back to folks within a day or two. We just like to make sure that we have all of the information specific to your question before we give an answer, just so that we can make sure that we're responding appropriately for your specific situation.
Yeah, well, I'll just note that, I know this is a complicated submission with pooling and sequencing and such, but, we've had it in for 6 weeks and haven't gotten any feedback, and I posed this question through the templates email and was told it would go into the queue, and that's been more than a week ago, so any guidance there that could get me a response, I'd appreciate it.
Well, I think coming in with a direct question of, and do I fall under the new policy for LDTs or not? And very simple. And if you don't get a response in 24 to 48 hours, ask to bring me, Tim Stenzel, into the conversation, or Toby, okay. And we'll get you a response absolutely as fast as we can.
Yeah, you can also.
We're trying to be patient. We realize there's a high workload there, so I will.
You can also specifically ask for something to be sent to me. It is very possible that it's on my plate since I was out last week.
Sure, no problem. Okay, well, thank you very much for the guidance. I really appreciate it.
Our next question comes from Daniel Roth. Your line is now open.
Hi, thank you, Dr. Stenzel and Toby, for organizing this. So we have been developing an antigen test and meant for home use. And we have 2 development paths as far as kit design. A colorimetric assay with enclosed containers, as well as a flow-through kit, also with enclosed containers. Originally, we had assumed that sort of the traditional lateral flow design, similar to an at-home pregnancy test, was a desirable kit design because it's fairly available. And thus far, from the sensitivity data we have, our colorimetric assay test is proven to have higher sensitivity. And so given that, and provided we put together a rigorous usability study with the protocol that you authorize, FDA authorizes before conducting the usability study, are you keen on authorizing under EUA an at-home antigen test with a colorimetric assay kit design? And secondly, are you keen to authorize a flow-through kit also with enclosed containers as a kit design?
Yeah, I'm not sure I know what the colorimetric is, this color line that disappears and or comes up depending on the function on the strip. What's the flow-through device? Is this just an electronic readout of some of the over-the-counter viral flow devices have a reader attached. I'm not sure what I understand by the flow-through aspect of your question.
Yeah. So
what do you mean by flow-through?
The flow-through kit design is nitrocellulose test strip where reagents are poured over and flowing through the test strip to enhance a visible change on the test strip that's visible to a human eye.
Yeah, no,
the colorimetric.
I was confusing the two. So by colorimetric, you mean on the spot where the sample is put down, there's a color change if the target is there, versus the flow-through strip where you put sample on one end and you read out down the strip after liquid has flowed through the device.
The colorimetric assay in the context of this question is without a test strip. It's a reagent that changes color in an enclosed vial, and this is a saliva-based test I should have mentioned. Yeah.
Yeah, so there's the three main things that we look for. So let me just, at a high level say, absolutely, we're totally supportive of authorizing home tests and home direct antigen tests. And in fact, I've noted on previous calls here that we haven't received a submission for a home test, whether it be molecular or direct antigen, that we can even review to authorize. So that's why I continue to encourage it. I've also let our staff know that I would be very interested in them authorizing a good home test as soon as possible. So they know when they see one, if it's good, that we want to do our best to get that authorized as soon as possible. I've looked at various developers for SARS tests in both these categories that you have talked about, and it doesn't really matter the readout method as long as it can be clearly understood by the home user and then the results clearly interpreted. So can they read the test as being positive, negative, or it didn't work, invalid, and then do they know what the results of that test mean? If they're positive, what does that mean? If they're negative, what does it mean? If they're invalid, what does it mean? And that the performance is such that we're not missing more than overall in whatever scheme you use. You could have two tests that they use, day 1, day 2, or day 1, day 3. That we are able to detect the vast majority of home users that are shedding virus that could potentially infect others. I usually talk about the first 5 to 7 days of shedding of virus in a newly infected patient. That may overlap symptoms or not. It doesn't really matter. That's what we're looking for. We want to see. We don't want to miss. We don't want to miss 50% or more, clearly, of people who could transmit the virus to somebody else. And we've set the bar at 80%. I think that's pretty good. It still means that we could be missing 20% of patients who could infect others. And going lower than that seems not the right thing to do. So, but we will work with developers if they have different schemes. So basically serial testing schemes are amenable if they're successful in getting that sensitivity PPA to a level that consumers and others can have confidence in a result. If it's negative, it's most likely negative. If it's positive, they're most likely positive. And that's really important. Okay?
Okay, thank you.
Our next question comes from Ted List. Your line is now open.
Hi, thank you for taking this call and for the opportunity to ask questions. My question pertains to EUAs that are for tests that require a physician prescription and a test that do not. And what are FDA's considerations when considering either, and how are they different with regard to the information that you would expect to receive from the developer?
Yeah, so obviously under prescription, we'll, everything essentially. I don't think we've authorized a non-prescription test yet for an EUA. And the reason for that is we can lower the bar a little bit when a clinician is involved, because that's a mitigation of risk. If they're involved, then they can assess the clinical situation and assess whether a positive sounds right or a negative sounds right, instead of a patient or a consumer being left entirely on their own and not having the relevant medical background to make an assessment. So when it's OTC, we generally expect the bar to be a little bit higher. So for direct antigen tests, for example, in an OTC where someone can just go to a drugstore and pick it up off the shelf, go to the cashier or self-checkout, go home and test and not have any interaction with a medical professional at all, we expect that the PPA or sensitivity for such a device is 90% for like a direct antigen test. But if it's by prescription, our bar of 80% is sufficient, because that additional loss of sensitivity, if the clinician's involved, we know that they can assess the situation and get repeat testing if warranted.
Okay, thank you for.
And this is also discussed in the home test template a little bit. There's some discussion about prescription versus non-prescription, because Tim was saying, it's important that for a non-prescription test, the user needs to be able to identify that that's the correct test for them to take. They need to be able to perform the test themselves and get the results and interpret the results themselves. So those are all factors that we look at.
Thank you.
Our last question comes from Christy Berkerson. Your line is now open.
Hi, Tim. Hi, Toby. Thank you for taking my call. So this is Christy Bergerson from Exponent Consulting's In Vitro Diagnostic Division, and we were looking on the FAQ page about dry swabs. And it says the shipping and stability data for the dry swabs was provided and accepted by Antigen, but the usability was not, and it needs to be assessed for each EUA. However, the next sentence after that says that the FDA still needs to review the clinical data on dry swab transport to understand the impact to test validity. Those sentences were last updated in May. Do you consider dry or direct swabs as an approved sample type at this point, or is the usability data the only data that's needed, or do you think that bridging studies would be necessary between wet and dry swabs?
So the limitation in that study, which Gates was also involved with, is that there wasn't a resuspension protocol of a dry swab. Whereas where the collection and transport of that swab data was provided there. Also, a universal sort of instructions for use wasn't provided with that and tested. The usability study is relatively easy to do. So the only other that I'm aware of that be made differently is that for a test, given a test that a dry swab is going to be used, is that swab adequately resuspended? You get the viral material off that swab adequately, and you get an accurate swab from that result from that setting. So that's the wet lab piece that's missing from that particular study, and so we do ask developers to develop and validate the resuspension method and make sure that they're getting good results from that dry swab. Toby, do you have anything to add to that?
No, that's exactly what I would have said.
Thank you. So does that mean that dry swabs are an approved sample type, or do we need the bridging studies between wet and dry swabs?
I'm not sure I can totally understand the question. You can add dry swabs if you validate the resuspension. There are different ways to validate that resuspension, obviously. But we are not requiring a bridging study to a wet swab for a dry swab.
Okay, perfect.
Each test would be authorized with specific sample types. And so if you're looking to include dry swabs in your authorization, we would want to see that validation.
Okay. Yeah, we need to understand the test performance if it's something that requires an EUA, and not just say, oh, we have a test and we don't have to test a sample type. No, this is one where, for something that requires an EUA authorization where an EUA authorization for some sample types has been issued based on actual clinical study data, positive and minimum positive and negative. And then if you're adding a dry swab, like a home collection to it, then that's where we don't need to see that clinical study data for that dry swab. We just need to make sure that the resuspension works. Good point of clarification, Toby.
Okay, excellent. Thank you all very much.
Sure. Thank you.